Thanks very much for coming. This is the 20th Paul Janssen lecture. I'm sure you all know who Paul Janssen was. He was the guy who invented haloperidol and set up the Janssen pharmaceutical company, who are the sponsors of this annual lecture. And the tradition is that we invite a world-leading investigator in the field of psychosis to give this lecture. We've had some very famous celebrity names in the past, and this year I'm delighted that we have Nikolaos Koutsouleris, who's a professor of psychiatry at the Ludwig Maximilian University in Munich. Many of you will know Nikolaos. He's been pioneering the application of imaging to psychiatric disorders, and particularly the use of advanced machine learning analyses. And a lot of that work has been done in collaboration with King's College here. And he's an honorary professor at the IOPPN, as well as having an affiliation with the Max Planck Institute in Munich. So Nikolaos is very interested in something that is close to our hearts at the IOPPN, which is precision psychiatry, and that's really going to be the theme of his lecture this evening. So he's going to deliver his lecture, and then there'll be time for questions at the end, although he's told me that you're allowed to interrupt him.
Sure.
So if you feel brave enough to interrupt him, go ahead. But if you
don't,
we can have plenty of time for questions at the end. So without further ado, welcome.
Thank you, Philip. Do you hear me well? Perfect. Thank you. It's really fantastic to be back to the IOPPN and to give this lecture today. I feel really honored, and it's also great to see friends in the auditorium and colleagues whom I met over the past years repeatedly, and we have projects together. Some of these ideas that have been developed together I will present today. The topic of the talk is, as Philip introduced, precision psychiatry. I will try to give you an overview of the state of the art, where we currently stand in terms of bringing this guy, to a more targeted, potentially, treatment. Assessment, first of all, differential diagnosis, potentially better prognostic assessment, and then, if that all works, in the future maybe to a more personalized, more precise intervention. The question is, however, if we use the term precision psychiatry, that implies that current psychiatry is imprecise. Yeah. I'm not so sure about that, actually, and I will give you some data, or show you some data today that actually shows that we are not as bad as we think. So the talk will focus actually on the idea of finding these, this is difficult to use, but anyway, on finding decision rules that allow us to bring a bit of a structure into the chaos of actually making psychiatry and assessments, diagnostics, prognostics more precise. And you see what the challenges are on that way. Let's start with the clinical scenario. So most of psychiatric disorders, as you know, or patients having different psychiatric phenotypes, there are subtypes across disorders actually that have a prognostic unfavorable course, who develop an adverse outcome as shown here. Many disorders have a prodromal phase in psychosis that's about 3 to 8 years, and then develop after this phase a full-blown disease phenotype. However, the problem is that this course does not occur in all of the patients, but it's highly heterogeneous. Actually, based on Paolo's work, and the paper, which is a bit outdated now, I think, we know that there are new data, and also from Paolo, Fusar-Poli, to be published soon, that show actually how unlikely it is to develop psychosis if you have at-risk mental state for that disorder. Actually, the current figures are, I think, about 20% of the individuals across the world who meet the CHR designation go on to develop a psychotic phenotype. And if you look at those who develop the disorder, this problem of heterogeneity continues. So we have here in this data from Gerd Huber's longitudinal 20-year follow-up studies, we have here a breakdown of the different recovery rates and courses in this population of 502 first episode patients. And you see basically, I don't know where I can get, here it is. You can see basically that everything is possible under the sky. You have people who completely recover after one episode, and on the other hand of the spectrum, you have patients who never get well again. And I think that's one key challenge that precision psychiatry set out to tackle. Let's see whether that will work out. Another challenge is shown here, and that relates to diagnostic conundrum in which we are in. Basically, there are fuzzy boundaries between the normative models that we have for the different diagnostic entities. Schizophrenia, for example, is here in terms of its reliability in this middle range, and depression is in the lower range, meaning questionable agreement of raters who determined the diagnosis in this DSM-5 field trials. Of course, that imposes a gradient of likelihood of detecting, let's say, neurobiological signatures for these disorders that we have to take into account when we try to build predictive models. And another challenge that I would like to show you here has been highlighted by the colleagues Hugo Schnack and René Kahn in their paper in 2016 that also illustrates the problem of heterogeneity. So as you increase the sample sizes that you use to detect predictors of, let's say, a specific phenotype, the more you buy in heterogeneity, complexity, because we don't have a gold standard, we just have silver standards, which is shown here. And the more data you have, the lower the predictive accuracies become. So the question is whether we, with this approach that is shown here of precision psychiatry, can tackle these challenges. That means by combining different readouts of psychiatric disorders, ranging from the clinical level down to the genome, whether we can decompose that heterogeneity in a longitudinal sense, in the cross-sectional sense, in the biological sense, in hierarchical from clinical phenotype to the genetics, and basically use that decomposition scheme to inform our treatment, which is shown in the lower part of the figure, to come up with more personalized treatments. I will focus on that part of this journey, actually. As I said, the approach is to use combinations of different data to understand heterogeneity here in this, let's say, patient population. Practically, for prognostic models, that means we project the data of these patients, this heterogeneous data, into potentially here a kernel space. This could be a Gaussian kernel as shown here. And we use an algorithm, be it a support vector machine or a random forest, to derive a decision rule that allow us to say with a certain probability that a given patient, let's say this guy here, has a high risk of developing, let's say, frontotemporal dementia. There are, besides these clinical challenges that I just highlighted, also methodological issues. And I would like to briefly make you aware of these issues. First, and most important, is the curse of dimensionality. Shown here, if you want to separate cats and dogs in a one-dimensional space, that's pretty difficult. So you want to add features, let's say the ears and the nose, the color of the nose. That is okay, but you're not happy with these models and you add further features. And as you add features, you basically have to grow exponentially your training samples to adequately model this problem, because the dimensionality tears these examples apart. That's one issue. Another issue that has been painfully highlighted in this publication story of the Drysdale paper, some of you may be aware of that paper. The colleagues basically published a paper on subtypes of depression that they identified with canonical correlation analysis. And they said that these subtypes are also relevant for response to RTMS. Here you see the two canonical correlates that they basically used to define those subtypes. And you see basically how strongly resting state patterns were predicting or explaining clinical variance. It's more than 0.9 Pearson correlation. And then the colleagues here, Dinga et al., published a replication paper which actually showed that you cannot replicate this approach. Why? Because the colleagues missed to do it properly. They didn't do a proper cross-validation. And when you do that, you see basically that CCA as a method always overfits the data and produces spurious correlation coefficients, which are most of the time greater than 0.9. And they did a permutation analysis here of this and found basically, just as I said, these are the permuted data correlation coefficients. Here, this the distribution. You see it's absolutely non-significant. So there are methodological challenges which relate to the curse of dimensionality and the way we validate these models. And if you bring this into a theory that has been described by Wapnick and Jawlonenski's machine learners who described the support vector machines in the 90s. If you bring this into the theory, VC, Wapnick-Jawlonenski's theory, you see basically with increasing power, algorithmic power to detect signature in the data or structure, you increase the probability of basically creating idiotic models that find the hair on the ear, that find some artifact that they use then to classify the specific sample. And if you take one of the most aggressive and powerful algorithms, which are wrappers, so these check each feature sequentially, using different search strategies. This is random data. You see this is basically the training data. It's 100%. You can always separate data, whatever data you have. And this is the test data. It's almost 50%. So that's a problem. If we try, the harder we try, put it that way, the more idiotic the prediction models get. And this is not something new. This is, if colleagues would have read about William of Ockham, an English medieval thinker, a monk, then it's clear that applying Occam's razor or lex parsimoniae would have helped. That means don't go for the most complicated model. If the models perform on the same level, choose the simplest one. And that's a general thing that I think people should use. We try in Munich to set up a platform that allows to address some of these weaknesses potentially, and that makes available different methods for standardized machine learning also that could be used to compare models and machine learning strategies, not only amongst our researchers, but also on a much broader scale. If you have a model, let's say, that is trained with this platform, we could evaluate it, we could test it in a different data set, and you can export it. And the nice thing about it is that it treats everything as part of a model process. So even the smoothing, for example, of structural images, everything that comes with a free parameter is basically registered in a heuristic space that is optimized. And a slide that I cannot hold back to show again and again. This is the validation approach. So you start with cross-validation, you go on with leave one side out validation, and you end with external validation. And this is also something that is implemented in that framework. So if you have a model, you think it's generalizable, you can basically enter a specific mode where you can ship it to another group and test it. And we are actually about to do that also in the Harmony collaboration, where also Philip and his team is involved in. So that's one important aspect. But finally, to really facilitate that validation endeavor on a large scale, I think it's important if you publish a predictive model to make it available to the world so that people who are completely independent of you, with no interest in your own career or whatsoever, can take the model and validate it. And we try to do that by setting up a platform where we register or we put in all the models that we want to publish. Here is one of the clinical models that has been published last year in JAMA Psychiatry on predicting social functional outcome in the clinical high-risk states. So these are more or less the theoretical. This is the theoretical part of the talk. I want to show you some data now, which tries to implement some of these ideas. The data comes from the Pronia sample. This is a multi-site European project that collected data in an observational study from clinical high-risk individuals and patients with recent onset depression and patients with recent onset psychosis. The transition risk in that population of clinical high-risk patients and first episode or recent onset depression patients together was 10.6% over a period of 2.4 years. You may ask, why the hell did they include depressed patients into that analysis? Usually people just look at the clinical high-risk state. And the reason is that we found that this depression group is quite close to the clinical high-risk individuals. If you look into the follow-up data, you see basically here in this stage 1A or 1B, so the clinical high-risk state psychosis, we see actually that there are 17% of depression cases who develop a clinical high-risk state during follow-up. And a small proportion, 2%, develop a full-blown psychotic disorder. That's a small number, that's true, but I think this illustrates that depression in that age range is different from depression when you're 40 or 50 years old. So they're relatively close to each other. And we have a lot of cases who have a little bit of, let's say, borderline symptomatology, OCD symptoms, they have depression, and they feel observed, sometimes. So you have these kind of diagnostic difficulties and chaos. And that illustrates that the conditions in that age range are still about to develop and crystallize to different directions. And therefore, I think it's important to leave the silo of the CHR state and look more broadly into related conditions. So we try to predict the onset of psychosis over this follow-up period using different data with the same approach. I don't want to go into technical details, but you see actually that with clinical data, you can predict with 76% accuracy, balanced accuracy. With polygenic risk scores for schizophrenia, just 10 features, you still reach 66%, and with neuroimaging, structural neuroimaging, you have 71% approximately. If we just look into the clinical data, these were the significant variables. More or less, what we see is that the patients who later on develop the disease are more severely affected already at baseline. They have childhood trauma loading, and they have some neurocognitive deficits in face emotion recognition. If we use, instead of a support vector machine, which was here the backbone for this, and a wrapper, we use in the wrapper, we use a simple univariate logistic regression. You see that the accuracy is lower, which indicates that indeed a multivariate algorithm is able to tease out a gestalt, a signature of emerging psychosis. However, I think it's important not to stop there, but really to try and to understand how does the model perform in other data? What has it learned? Is this an abnormal pattern, abnormal from normality? And we try to investigate those questions. First of all, the external generalizability of the structural imaging predictor. We use two different samples, both from Switzerland, one from Basel and one from Zurich. We can of course argue these are close to each other. This is not a good way of assessing global scalability, but this is maybe a start. Yeah? So you see that in completely unseen data, the structural pattern, the signature, replicates with 68.5% balance accuracy and 69.3% balance accuracy in samples with completely different transition risks. So the Zineb sample is very close to the Pronia sample, whereas the old FAPSIS sample has much higher transition risks. So that's interesting that this model, although trained in a highly unbalanced scenario, very few transition cases, a lot of non-transition cases, still performs well with different risk enrichment levels. What have we learned? Is this a deviation from normality? Here we see here a comparison to healthy controls, and we found that in these blue areas, the cerebellum and posterior hippocampus, patients with a transition have more brain volume than healthy controls, and the opposite is the case for the red, for the occipital areas. And for the non-transition cases, the pattern is reversed. So it's indeed, in both directions, a deviation from normality. We don't know what it actually means at the moment. Let's have a look at the polygenic risk score predictor. So here we try to understand the same thing. These are the polygenic risk scores, so the thresholds, the genome-wide thresholds, from very conservative to lenient to p equals 1, actually. And you see basically that after this threshold here, you get also in both directions significant differences. So transition cases have higher polygenic risk scores. Non-transition cases have lower polygenic risk scores compared to healthy controls. And that opens up a box of the discussion, are polygenic risk scores not only a risk factor, but also maybe a resilience factor if you have lower against psychosis. So do we get better predictions if we combine data? We tested that using stack generalization. This is a way of fusing information across different classifiers. That was properly cross-validated. So the single or unimodal predictors were concatenated, combined in the same training folds and tested in the same test folds. And we see here that the clinical model is the most important component, and it gives a boost of classification performance from 75.7 to 83%. Then another important question, which now brings the whole story into this area of clinical implementation. Do we gain something from compared to clinical raters when we use all these algorithmic engines? Or does it make our life more complicated? We asked our colleagues to make a prediction after the baseline, and surprisingly, and this relates to my initial question, imprecise psychiatry, they were not that imprecise. 73%. That's as good as the unimodal classifiers. And interestingly, look at the sensitivity and specificity. We are very good in ruling out transition. We are not very good in doing the opposite. So it's a biased pattern recognition algorithm that runs in our heads, obviously, or at least in those young people who assessed these patients. Maybe that's reflective of an optimism bias. So that's significantly worse than the combined predictor, but it's not rather man versus machine, but the question that actually replicates what we found in the social functioning prediction analysis published last year. So adding data is better than each model alone and better than the raters in a different domain. But when we integrate, the question is, do we gain something in addition? So we treated the raters as a decision stump model, did the same thing, and we found that the raters now are the most important component in the stacked classification algorithm, and we get a little bit more. We get now up to 85.5%. And my beloved imaging is not so important, so I tested that. It's beloved but very expensive. If you remove neuroimaging data, you still get 84%, which means there is redundancy among the components, which is a good thing, I would say, because you can adjust these pipelines to different clinical settings, even to settings where no MRI is available or too expensive. So, but for clinical applicability, we don't have the data all available from the beginning when we see the patient for the first time. We would do it sequentially. We would do first a clinical examination, maybe then potentially an MRI scan for exclusion diagnosis to exclude encephalitis potentially. And then we would do maybe a polygenic risk score. So the question is whether by strategically combining data, we can, for this case, which is ambiguous, here you see the circle, we can achieve a strategic shift towards one or the other class and boost our prediction performance. We tested that. This looks like a messy graph for for these different modalities, including the raters. And we found that among all the tested sequences, 270 different combinations of parameters, there are actually only very few that are really good. And the best one is shown here. We start with clinical data, proceed to polygenic risk score data, add imaging, and at the end, we ask the raters what they think. Yeah? And because the raters are very specific, that helps to rule out false positives. And that's actually what you gain here. So the combination of data in a strategic way. If you have been labeled with a likely transition, you have to travel the whole predictive journey, sort of. Okay, that's the prognostic application area. What about differential diagnosis? So when you train a classifier, you can actually span a similarity space. It's not yes or no. It's rather shades of gray in the, that the classifier can measure. And you can use this strategically to study another condition. Let's say you train a classifier in schizophrenia. You apply that to depression to investigate whether the signature of schizophrenia is present in this other condition. So encapsulating a disease signature and delivering it to another field. This is what we did back in 2015, exactly on that question. This is the classifier differentiating schizophrenia against depression, and then we used it to label this, I would say, sort of intermediate phenotypes. So this is bipolar disorder, and these are at-risk mental states and first episode psychosis, and thus testing the old idea of the Kraepelinian dichotomy that you have manic depressive illness on the one side and dementia precox on the other side. And interestingly, we found that there is a distinction. Yeah? So bipolar disorders are labeled as depressed patients by this classifier, whereas the at-risk mental state patients are already having the schizophrenia signature in their brains to different degrees and different variability. Let's return to this notion of heterogeneity. Actually, Kraepelin was particularly describing, I would say, these cases that we call today Kraepelinian cases. There are hypotheses that those patients, they follow on this neuroprogressive path, on a specific neuroprogressive trajectory of this disorder. It's a hypothesis that is popping up again and again over the last 100 years, I would say. And that is controversially discussed. Maybe machine learning could be an avenue to test this on a systems level approach. As you know, patients with frontotemporal dementia, when they are particularly young when they develop the disorder, their phenotype is quite close to schizophrenia. We also say this is like a phenocopy of schizophrenia. And this has been systematically assessed by Dennis Velakoulis, and you see this relationship that I just told you about here in this figure. So we set out for a project together with Matthias Schröter from the MPI of Human Cognitive and Brain Sciences in Leipzig, and tested that hypothesis that there is a continuum between the disorders systematically. So here is an FTD classifier on structural imaging data. You see this clear pattern of frontotemporal volume loss in the brain. And that performs with 86% accuracy. That's Alzheimer's disease from the same consortium. It's a different pattern. You see that here the hippocampal limbic regions are particularly important in the precuneal regions. And that's schizophrenia from our Munich cohort. And that's the most extended pattern and the lowest accuracy, which reflects the notion that schizophrenia is a heterogeneous disease, so the classifier needs a lot of regions to come up with a decision boundary. What happens if we now apply these two models, FTD and Alzheimer's disease, to the schizophrenia cohort to label their FTD versus AD likeness? So we see that the patients with schizophrenia, or some of them, actually 30%, are labeled as FTD patients. That's not the case to that extent for their Alzheimer signature. So that there seems to be a preferential loading or a neuroanatomical proximity to FTD. Then we wanted to understand whether that FTD signature in schizophrenia is related to symptoms. Yeah, and we trained a support vector regression to predict FTD likeness using clinical data and brain age, so accelerated aging in schizophrenia. That works quite well with variance explanation of 43%. It works less so for AD scores. So you can predict the FTD likeness of schizophrenia patients with their clinical phenotypes to a certain degree. And what's the clinical landscape that is associated with that? It's brain age, of course. It's the body mass index, which might reflect disease, so treatment effects, but it might also reflect disinhibition. Yeah. It's illness duration, and its grandiosity, and lack of spontaneity and flow of conversation. Items that align quite well with the FTD pathology, I would say. Then we took this FTD classifier, and we were interested, is this only relevant for schizophrenia, or is it already relevant for the clinical high-risk cases? That's a bit of an audacious question. Yeah? So you're relating a pattern that is developed in 60-year-olds to brains of 20-year-old patients. So you can do this by normalization procedures, taking out the multivariate signature of normal brain maturation and aging. And when you do that and relate this to the 1-year functional outcomes of these clinical high-risk individuals and recent onset depression patients, you see that in these Frog's dimensions, yeah, and the GUF symptoms, there is a significant effect. So patients who are basically have a higher FTD score have lower functioning after 1 year across almost all these domains. And that's driven by the clinical high-risk individuals, not by the depressed patients. So it seems that already in that prodromal or at-risk state, having a loading on this FTD pattern predicts poor outcome. That's not the case for schizophrenia, for the schizophrenia pattern applied to this early cohort. And it's not significant for the Alzheimer's disease pattern. And we turned the story around. We trained a classifier in the CHR and ROD cohort. And this is new data that will be hopefully published soon. We trained it to differentiate between really, really impaired patients over time, so using a GUF disability score of lower than 51 over all time points versus not. And we then, that works actually not so well, it's about 60%, but if you apply it to the FTD cohort, you see that it works as a very nice classifier. And that is, for me, I think, a striking finding. So that means these signatures, they have a lifespan and a dimensional perspective that covers, it embraces not only FTD, but also subtle conditions like the CHR case. Yeah, normative modeling. This is the third application area that I wanted to show to you. I'm looking at the watch, still a little bit of time. Another approach to understand abnormality is using healthy controls or epidemiological samples like the UK Biobank, for example, or other samples to set up models that allow us to really quantify and measure abnormality in a multivariate sense. A simple approach to that is abnormality across the age dimension, and this is nowadays commonly referred to as brain age concept. We did that, and then you basically, you use this model to understand abnormality, for example, in schizophrenia. We did that a couple of years ago in a then quite large data set. Now we have much larger data sets and nicer papers out which actually replicate that finding. You can very nicely predict the age of an individual, of a healthy individual, across the lifespan using neuroanatomical data. The error is about 4.5 years. And when you apply that agnostic model, normative model, to patient populations, you see that there's systematic effect of misprediction or wrong prediction. Why? Because in schizophrenia, which is shown here, you have brains that look older than they should look like. So, And that is something that is not specific for psychosis. It's present basically to different degrees also in other diseases, such as major depression, for example, with a strong or high variability. And is that brain age? Is that relating to our ability to classify and discriminate at the brain level? We investigated that by comparing the classification approach that I showed to you previously versus the normative modeling approach. And we see actually that and the brain age is also a good classifier, yeah? Differentiating patients from controls. And actually these two signatures, a classification model trained specifically to do so, whereas an age prediction model broken down into a classifier. They strongly overlap, here shown in yellow, and the brain age model explains more than 50% of the variance of the classifier. That means in the end, if we use structural imaging data, what we most of the time, or the largest part of the variance that we use is accelerated aging related or brain age related. And that informs also our differential diagnostic model that is shown here. I think it's important to stress again and again how crucial it is to understand model performance and what effects drive this performance. And here very clearly, brain age was driving the separability of depressed patients. So the younger the patients were, that is not shown here, but the higher the brain age was, the more likely they were classified as schizophrenic patients. So you see how these differential diagnostic axis and the axis of brain maturation basically intersect, yeah, in a high dimensional space. And that is just one of the possible axes that influences classifier performance or predictor performance. There might be many more which are completely unknown, and I think we need to systematically set up such normative models to better parameterize what models actually do. And that's really crucial if you want to bring this into the clinic. If you don't do that, these models will have a very, very restricted application area. And you will never know whether you predict transition to psychosis, or whether you predict accelerated aging, or whether you predict FTD maybe in 30 years. Final approach, clustering. As I told you initially, heterogeneous disorders, we have to deal with those in our field, and one way to decompose heterogeneity and potentially better predictors is the clustering approach. So one colleague of my lab, Dom Dwyer, did an analysis again with structural imaging data. I'm repeatedly referring to that domain because I think it's interesting how you can look at these data from different sides. So he tried to understand heterogeneity in schizophrenia through this lens of clustering. This is a model without clustering. And you see that you can classify healthy controls from schizophrenia patients with an accuracy of 68%. This is the pattern in red areas, volume reductions. Actually, this is the familiar pattern that we always find, and that I also showed you in the volumetric analysis previously. When you combine this with a k-means clustering algorithm, so you subtype the schizophrenia cohort, we found that there are two subtypes. He called it brain type 1 and brain type 2. They look quite different. And when you do that and classify those against healthy controls, you get an increased balanced accuracy. Why? Because heterogeneity has been decomposed and resolved. And it's not basically biasing the model or to that same degree as in the full sample model. The important thing again, now again back to the methods, is to externally validate that. Because as you saw with the canonical correlates analysis, you can find all sorts of spurious associations. So he did that, taking the Cobra sample as a discovery sample and applying the clustering solution to the Munich data, and he could more or less replicate it. Yeah? So particularly in this brain type 2 classifier, he found an increase in classification performance. And that leads me to the conclusions of this talk, and then we have a little bit more time for questions. I think to move this field and this new field forward, we need to really standardize our modeling tools. It's not a plaidoyer for our software, but rather for standardization that is really, really urgent, yeah, if we want to achieve progress towards clinical translation. I think it's important also to leave the silos in which we all work and to look at the different phenotypes in a standardized manner, following a comparative neuroscience approach. Looking at conditions which have phenotypic similarity with our main interest phenotypes, let's say psychosis, FTD. Could be other conditions, dementia and major depression as well. You know, major depression, it can be a prodromal state for Alzheimer's. And I think this could be a pathway to better deconstruct heterogeneity and thus help build better models and at the same time better taxonomies. I think it's important to properly cross-validate and validate. Leave side out is one approach that is also now increasingly favored in the literature because it provides good estimates of the geographic generalizability. Actually, we see that in the Harmony collaboration that you can leverage large cohorts which are diverse, and then you can nicely establish measures of out-of-sample performance. And what's needed for that is moving from a more competitive towards a more collaborative, large-scale, multi-site and diverse patient sampling approach. What I could show to you today is that neurobiological data doesn't seem to offer any superiority over clinical data. That's interesting. What we find, though, is superiority of combined or multimodal predictors over unimodal, including raters, unimodal models. Yeah? So, and I think one of the last steps is to evaluate validated models in risk-stratified clinical trials. And Philip and Paolo, they are leading such an effort with the STEP project. And I think this could be a way forward in the future to test whether these models are really clinically useful, yeah? Or whether it's just la polar that we do. We don't know. Yeah? It's not man versus machine, but man and machine. And finally, it's important not just to look at algorithms, but also at humans and what drives pattern recognition in the human brain. What are the biases, and to better understand that in order to build better algorithms that actually enhance it and not counter it. Okay, thank you. That's it for me. I'm open for your questions.
Okay, Nikos, that was fantastic. So we have a bit of time for questions. I'm going to take my privilege by asking the first one. So I was slightly horrified when you showed the data that neurobiological measures added nothing to clinical prediction. So can you just talk us through that? Because some of us have invested our entire career.
Including me.
Including you. But globally, people are throwing billions of euros and dollars at neuroimaging prediction.
Yes.
So what are the implications?
So in that scenario with the three modalities, clinical, polygenic risk score, you have a neurobiological data domain and raters. It's basically on par with the full stacked model, but in other scenarios, not having a biological domain, just clinical data and raters is significantly worse, that's 77%. So having one biological domain seems to help. Why? Because these domains are very sensitive. And genetic and data or polygenic risk score data in that setting or neuroimaging that might be replaceable. Yeah? So you might just need one, but they add. Yeah? They add definitely. I don't know whether we need imaging though, for every setting. And maybe that's in that scenario like that. In other scenarios we need imaging and it gives a strong boost. Yeah? But that's what we measured. I mean, that's it. Yeah? Okay, so we have a microphone. If you have any questions, please go ahead. And yes, Paolo is down here.
Thank you, Nikos, very much for your brilliant presentation. I've got a related question. One of the take-home messages is that multimodal algorithms are better than unimodal algorithms. At the same time, they are more complex.
That's right.
So what's the trade-off between implementation of those models in clinical routine, and there is some tension? Yes.
For clinical care, I would recommend not to go for these kind of fully stacked models, but rather to implement these sequential prognostication algorithms. And they are also, from a health economics perspective, much more viable, because you don't need to test all the patients. For this example here, for the predicted transition cases, yes, you would need all the modalities. But for the non-transition cases, you can stay just with the clinical data. And that's, I think, to have this differentiation and the flexibility to combine data, that I think helps. And the other thing is a methodological perspective. We tested that systematically, and it seems that this decision-based data fusion, so where you learn a multimodal predictor from the output of the unimodal classifiers, is better than putting all the data together and learn one model. And that might also be the reason why, for example, deep learning has this edge over more traditional machine learning algorithms, particularly in large sample scenarios where the complexity isn't the problem anymore, because you can represent the full space even if people basically would be far away from each other due to the dimensionality. You can fill the gaps with the data and thus learn, even with high complexity algorithms, well generalizable rules. But I think this decision-based fusion makes a difference.
Thanks very much. That was a fascinating talk, particularly exciting about the predictive capabilities and clustering of subtypes and so forth. But I have a question about translating to clinical care in particular, and whether or not your group or you're aware of research that have looked at precision psychiatry in response to treatment or types of treatment, if there are certain characteristics that make a certain individual would respond better to different types of treatment.
Yes. So we have two papers on that. One is on RTMS, outcome prediction, in chronic schizophrenia. And there definitely you can also, in a leaf side out fashion, when you validate the structural imaging algorithm, you can predict who is going to respond to this intervention or not in patients with predominant chronic negative schizophrenia. So that's an intervention on the dorsolateral prefrontal cortex, specifically targeting negative symptoms. Another area which is interesting is a clinical model that we trained on the UFEST sample. This is the first episode sample, first episode psychosis. There we tried to predict functional outcome after a year, and what we found there is that the patients with a predicted poor prognosis, predicted, not observed, predicted poor prognosis had differential response likelihoods. That was not the case for the patients with a predicted good outcome. Yeah? And that means that basically these clinical trials are swamped with patients who will respond anyway, and therefore the effects are diminished that we measure. And in this cohort, which has the predicted poor outcome, we found that amisulpride was far better than, for example, quetiapine. And it was so good that it was beating the predictions of the algorithm in 80% of the cases. And the opposite was true for quetiapine. That's all post hoc, but that's a hypothesis that you can formulate based on a machine learning analysis that you can take then into clinical trial.
It was very interesting. So I'm interested in the brain age measure.
Yes.
And the extent to which that takes into account the propensity of people with schizophrenia to have early obstetric and other insults, to smoke excessively, to be obese, and to be plied with antipsychotic drugs over many years.
It doesn't take it into account. It's a very dirty measure in that sense, very unspecific, because all these effects that you were just referring to project on this coarse voxel level measurements that we use. You would need to train submodels where you have basically measured all these influences, these moderators, and decompose the factors that influence that model. That's possible, but you need to have the data for it. Yeah?
But to some extent, you extrapolate from the brain aging as part of a construct of the illness as a progressive. But maybe it's the things that happen to psychotic patients that are progressive.
Yes. And the FTD results would actually illustrate that, and because there the FTD score was most predicted by brain age, yeah, and also the illness duration and the negative symptoms and grandiosity, and that shows that there is a subgroup of patients who might be on this neurodegeneration trajectory that Kraepelin was initially thinking of, dementia precox. Yeah, I remember your discussions with René Kant, which were actually always really, really good, where you had this nice dispute. Is it a progressive disease or not? Actually, it can be both. It's a heterogeneous disorder. It has the possibility to develop in these 2 or 3 or 4 directions. And one direction is related to this neuroprogression hypothesis, I think.
Hello. Yep. Thank you for the talk. How do you go by implementing this, asking the opinion of the clinician? Because in the case you presented, it's very clear, but people could be thinking that just their opinion are valid, so the algorithm is giving nothing. And
that's a good point. Actually, here it shows that the rater's opinion at the end of the prediction pipeline is the best, but that would mean for practical implementation that you have to hide all the results produced up to that point from the raters. Because in Pronia, people were asked without having all the knowledge. They were just having the knowledge from their interviews with the patient. You know? So, and that, importantly, that was a procedure that Netto took about, I would say, 10 hours. So they had much more time than a normal clinician had. And the question is, of course, if we want to take this into the clinic, what's the performance of a normal clinician? I don't know. Yeah? But I can say that if you're trained and you spend enough time with your patients, you are a reasonably good classifier.
Hello, I forgot what was mentioned there, but what was the false positive rate there? And wouldn't you be worried with the clinical implementation of the over-medicalization of the population, as well as of the fear of illness?
So the false positive rate is concerned.
Yes,
Yes, it is, definitely. And you see basically that both the imaging and the polygenic risk score-based models, they had a specificity of 53, 57%. Basically almost half of the population was labeled, of the non-transition cases, was labeled as transition cases. You cannot afford that. Yeah? That's so the implementation is all about managing these false positives. This is what I think after seeing these data. And clinicians, at least the Pronia clinicians, were good in bringing down that rate. There might be other ways of doing so. Yeah? But I think it's all about having a very sensitive model in the beginning, which may even create a lot of false positives, but then you don't miss anyone, and then bring down the false positive rate sequentially.
I just think at the moment, maybe for a decade or so, it could be more use of understanding models of psychosis and looking for solutions, psychopharmacology, rather than in the clinic.
So what would be the question?
It would be better maybe used for looking at prediction models, looking for the models of psychosis and targeting them with pharmacology in the lab rather than for the clinical implementation.
Yes, yeah, that's also, if I understood you correctly, that could also be an application area where we subtype different response types based on maybe the neurobiology, using these kind of approaches, and use this to inform, let's say, treatment and clinical trials and so on. Yeah, yes. Yes.
So Nikos, bearing in mind all this discussion, how far off are we for, or do you think this is ever going to happen, that there'll be some kind of tool on a mobile device that a clinician will be using in the real world?
I don't think we are far away.
So give us you'll be a record on camera. How many years?
Well, this is a difficult question. I would refrain from making an exact estimate, but I think if we look at the prediction models that are now validated, including the work that has been done here, and also the Naples risk calculator model, that's pretty close to be tested for clinical utility. Whether it helps, that's unknown. Yeah? But I think we are in the phase where we can use models that you can at the moment count maybe on your two hands. You can use those models in a clinical trial phase. So that would be, I think, the next stage for this field. And that, yeah, would take several years, obviously, to test.
Wonderful answer. Okay, so I'm going to invite Paolo Fusacoli to give us a vote of thanks.
Thank you very much. It's my pleasure just to thank Professor Koutsouleris for his inspiring lecture and for sharing your vision in this field. So let's just... I was told to keep it brief because there is a wine reception on the first floor, and thank you all for coming tonight.